Today's topic of discussion is acid fast staining. Okay. First question which arises is why it is used for, what organism can be stained using this. The thing is that it can be used to identify acid fast organisms like Mycobacterium tuberculosis. These are some pictures. Okay. Now we will get a question. How can you say that an organism is an acid fast organism? The organism which takes up the stain is called as acid fast bacteria. We use a stain in this technique called as carbol fusion stain. And when we discuss about procedure, I will clearly explain why it is used for and how long we should use it. Okay. Now just get to know a stain called as carbol fusion is used. The acid fast bacteria, if they have mycolic acid, a component in their cell wall, then they will take up this stain and will turn out to pink color. Okay. Only if they have mycolic acid, they will take up the stain and turn out to pink color. Those organisms is called as acid-fast bacteria. And the best example is Mycobacterium tuberculosis. Let's get deep into the topic. There is a technique to perform this acid-fast stain called as Ziehl-Neelsen technique. It is also called as hot method. There are four important steps to perform this. Whenever we have to perform a stain, we need to prepare smear for shear. So, smear preparation. We have to do heat fixation, reminded. There is a three-step procedure in this. And finally, interpretation, which will help us to find out which is an acid-fast bacteria. Right? Let's get into the topic in detail. Now, smear preparation. You have to prepare a smear on sizes 2 cross 3 centimeters. Okay, then we have to take a sputum, and in the sputum, yellow purulent portion must be taken. What portion? Yellow purulent portion of the sputum must be taken, and the sputum must not contain bubbles and all that. Okay. The next thing is that what we have to look in the slide is the slide must be grease free and scratch free. You should not take a scratched slide or which is greasy. Okay. Then an important step, note this out. We have to perform this near a flame because there is a rule that 6 inches around a flame is said to be sterile. Is said to be sterile. So to make this procedure sterile, it is better to perform this procedure, this smearing around a flame. And another theory is that heat will coagulate all the aerosols erased from this smear. With this theory, we will get into the second step of this preparation called as heat fixation. Now we have a smear which is 2 cross 3 centimeter in size. We will air dry the smear for some time and then we will pass the smear over a flame and we have to pass it for 3 to 5 times up. Every time you have to wait for 3 to 4 seconds. Okay. Show the smear in the flame for 3 to 4 seconds. Take it off. Then again take it off. Do it for 3 to 5 times up. Why do we do this is that the proteinaceous material, already I said it right, heat will coagulate the aerosols. What it coagulates is that the proteinaceous material will be coagulated, right? Once this proteinaceous material is coagulated, the smear will be fixed. Our work is done in preparing and fixing the smear. Now we can take it forward for procedure. Now, the procedure has three steps. We will use a primary stain, then we will decolorize it, then again we will use a counter stain. Okay, very easy. You're using a primary stain, then decolorizing, then counter stain. Let's see what are those steps. First step, the primary stain that we are using is called as carbol fusion stain. You have to take 1% carbol fusion stain it for 5 minutes. Okay. Some people, for better penetration, we may do intermittent heating. But remind one thing in your mind, though you do intermittent heating or you may not do it, you have to keep one thing in your mind, that is that the smear, the slide should not get completely dry. It should not completely dry. When you rinse it off, the free carbol fusion must be rinsed off and not the fixed ones. Only then you will get a color red in that smear, in that slide. Okay. If you get once you rinse off, if you are getting the red color, it means that you have done it correctly. Second step is that you have to add sulfuric acid, 25% sulfuric acid. First step we waited for 5 minutes. Second step we are waiting for just 2 to 4 minutes up. Again you will rinse it off in the tap water and now tilt it and air dry it. Okay. This is a decolorization step. We are using a decolorizer called a sulfuric acid. What will happen? The stain has to be colorless. If your stain is still red or pink in color, reapply, reapply. Okay, what? The decolorizer and wait for 1 to 3 minutes again. Nothing wrong. Okay, and once you get this colorless stage, you can proceed for next step. Done. The third step is that we have to use a dye called as methylene blue, which is 0.1% and wait just for 30 seconds. Okay. Again, rinse it off. Finally, you're getting a slide. You will have a slide with you. Take that slide and weave it under a light microscope. First, you have to fix the field, right? You have to get a suitable area. To select the suitable area, you have to weave it under a low power objective 10x. Then we will see it under oil immersion field 100x. So if your slide is once used or you have contaminated the slide, sometimes it happens, okay? If it is contaminated or you have used the slide, you have to dispose it properly. You have to dispose it in a 5% phenol solution. Note this point. You will be asked in your Viva sometimes. And then how can I interpret this? I did all this step. I got a slide in my hand. How can I interpret? Very easy. You will get long, slender structures in your microscope. Those structures will be straight, sometimes slightly curved or beaded. So you can see everything beading, straight, slightly curved, right? And the structures will be red in color. And the background and non-acid fast bacilli will appear blue in color. This background is blue in color, right? Others will be blue in color and the stained bacilli will be red in color. Actually, it appears pink, but when you see it on microscope, it will be red, right? So that's it. Subscribe, guys. Thank you.